Hello, Life Science. Hope you're doing well. Today we're going to continue Chapter 6, Role and Components of the Immune System with Document 6, Antigen Recognition by B lymphocytes. So we are done with Document 5. Let's do a quick recall before I continue in Document 6. So we saw in Document 5 that the main lymphoid organs are primary lymphoid organs, bone marrow and thymus, and secondary lymphoid organs, lymph nodes and spleen. The bone marrow and the thymus are responsible for the production and the maturation of lymphocytes B and T, and the secondary lymphoid organs are the site of action of lymphocytes against antigens. We saw that mature LB are lymphocytes B whose receptors can recognize and bind only non-self antigens, and the recognition is done directly. Recognition of the antigen is done directly, while the lymphocyte T, their maturation happens in the thymus, and LT mature are those having TCR that recognizes indirectly the antigen. What does that mean? Lymphocytes T have TCR that makes a double recognition, a recognition of the self HLA and a recognition of the non-self peptide carried by the self HLA. So the T lymphocytes, they recognize the antigens actually that are intracellular that infect the cells. Okay, now we can start with a new document, that is document 6, which is the antigen recognition by B lymphocytes. We're going to go more into details about the structure of antibodies and how do these antibodies recognize and bind to the antigen. So, antigen recognition by B lymphocytes, it occurs directly through BCR or antibodies. So what is the chemical nature of these antibodies and what are they characterized by? The structure of antibodies. What are antibodies? Antibodies are Y-shaped proteins. They are made up of 4 chains of polypeptides. 2 heavy chains represented in blue in the figure, the long blue chains, and 2 lighter chains also in blue, but they are the short blue chains. They include a constant region C and a variable region V. So if you look at the figure, you see the constant region is the region actually that is constant among all antibodies. And V, the variable region, it's the region that includes the antigen binding site, and thus it varies according to the type of the antigen which is represented in yellow. So that's why we call it the variable region. It differs from one antibody to another according to the type of the antigen, and it's this region that allows an antibody to recognize a specific antigen and to bind to it. Also, you can see that the constant region also possesses an effector region that is responsible for the fixation to certain membrane receptors. What is the role of the variable region? So the variable region is responsible for the recognition of the antigen. Actually, every lymphocyte B produces only one type of antibodies. So you have here, for example, a B cell, a B lymphocyte presenting one type of antibody represented in blue, and this antibody is directed against the mumps virus, and another B cell producing an antibody against measles virus, the antibody in orange, directed against the measles virus. So the anti-mumps virus can only bind through its variable region to the mumps virus. And you see that the anti-measles virus in orange are unable to bind to the mumps virus. Similarly, the anti-measles, the anti-mumps virus in blue are unable to bind through their variable region to the measles virus. Another example here shows the role of the variable region and the specificity of the antibodies. So the first antibody binds to an antigen that is compatible with its antigen binding site. The second antibody in yellow binds to another antigen also that is compatible with its antigen binding site. You see that every antibody carries two antigen binding sites that are identical. So the first antibody has two identical antigen binding sites and the same for the second. What about the constant region of antibodies? So the constant region, it presents slight variations that determine the different classes of immunoglobulins. So, according to the constant regions, we can see that there are 5 classes of immunoglobulins or of antibodies, known as IgM, IgG, IgA, IgE, and IgD. So, the figures below represents the structures of the 5 immunoglobulin. And IgM, for example, is a pentamer because it's formed actually of 5 antibodies, of 5 immunoglobulins bound to each other. The IgG is a monomer. The IgA, for example, is a dimer made up of 2 IgA antibodies. The IgE and the IgD are also monomers. The difference between these 5 types of antibodies consists mainly in a difference in the constant region. So the constant region is constant in the same class. For example, if I'm talking about IgM or a class IgM antibodies, so all IgM have the same constant regions, while if I compare IgM to IgE, for example, we're going to find that there is a difference in the constant region. So from class to another, the constant region may vary, while in the same class, it's the same constant region. Okay, what do we mean with the specificity of the antibody? An antibody recognizes only the extracellular antigen, that is the antigen that does not infect our cells or the antigen that stays outside. And that's what we meant with direct recognition of the antigen. So the antigen doesn't enter the cell and it won't be cut into peptides. It won't be carried by HLA on the surface of the body cells. So the antigen is outside. It is directly recognized by the antibody. So the antibody recognizes this extracellular antigen, whether soluble or cellular, and binds to this antigen in a specific manner. How? By binding to an epitope of the antigen. So we used to say that the antibody binds to the antigen. Now we're going to be more specific. Actually, the antibody binds to an epitope of the antigen. So what is an epitope? The epitope is called also antigenic determinant, It corresponds to a fragment of the antigen. So the same antigen can have many epitopes. As you can see here in this figure, the antigen in the center has, for example, 4 epitopes represented on the right side of the slide, the 4 types of epitopes. And you see that every epitope is bound by an antibody. So you have antibody 1 in yellow binds to an epitope. Antibody 2 binds to the other epitope. Antibody 3 and antibody 4 also, they bind to their specific epitopes. So you see that the same antigen having 4 different epitopes can be bound by 4 different antibodies. So I can't say anymore that the antibody binds only to one antigen. Actually, the antibody binds to an epitope that is a fragment of the antigen. So, note the antigen binding site of the antibody and its complementary epitope. They have complementary shapes and fit together like a lock and a key. They bind to each other through a non-covalent bond. The last part in this document is the cross-reaction. What is cross-reaction? It is the ability of an antibody to bind to 2 different antigens. How come? We know that an antibody is not specific for an antigen, but rather for an epitope. So how come the same antibody can bind to 2 different antigens? So whenever these antigens have a common epitope, the antibody can bind to these 2 different antigens. So we say here that the antigens cross-react. Let's see an example. You have below an anti-A antibody that is specific for an epitope of the antigen A. So actually, this anti-A is called so anti-A because it's directed against an antigen called antigen A. So we call the antibody anti-A antibody. Okay. But how come anti-A antibody in the second figure is able to bind to another different antigen called antigen B? Is it because they have a common epitope? So, the antigen B, as you see, carries a common epitope with antigen A, represented in blue. That's why anti-A antibody can bind also to antigen B by binding to the common epitope. So we say in this case that these antigens A and B, they cross-react because the same antibody, anti-A, is able to bind to antigen A and to antigen B because these 2 antigens have a common epitope. Another example is that of your book in document F, the cross-reaction. You see here the same antibody directed against the epitope Z, knowing that Z belongs to 2 different antigens. So this is what we call the cross-reaction. Thank you for your attention. See you later.